do a genetic screen. And probably many of you have either done one or thought about doing one, but there's lots of reasons you might consider doing this type of thing. I'll describe partly by way of historical context, but partly by way of describing how exactly it is that the Cas9 system has been so powerful at the functional genomics scale, comparison to some RNAi technologies that were used early on in functional genomics. And in my own lab, we've done some direct comparison between RNAi and CRISPR that actually is somewhat illuminating in terms of understanding the difference between a knockdown phenotype and a knockout phenotype and what you can do with that information. I'll describe a number of different screens that have been done by a number of different labs. I'm going to focus some on the screens that we've done just because I can talk in more detail about them, but I'll describe some particular applications in terms of identifying the targets of different drugs and phenotypes for essential genes, which are very easy phenotypes to screen for. I'm going to describe a little bit a continuation of what Luke set up and using CRISPR-INA to do genome scale screens in really elegant work that's been led by Luke and Max and others in the Weissman lab. And throughout, I'm going to sort of try to give actually some tips and things that are sort of practical things that we've learned in the process of doing a lot of screens, as well as different design considerations that you might consider if you're thinking about setting up one of these screens. And then in the last bit, I'm going to describe two relatively new applications of CRISPR screening that we've been exploring in my lab, but have also been used in a number of other labs, and in particular, genetic interaction screens where you're knocking out pairs of genes to look at synergies between different pairs of genes, and then a scaling of the technology that Luke described at the very end of his talk, which is what can you do if you can mutagenize a large stretch of DNA. And then at the end, I'll just describe some useful things for what do you do when you have a screen, the results from a screen, how can you actually use that information. So just one really basic slide, I think, to kind of understand where screens have come from. Probably this may be too basic for some of you, but before the sequencing of the genome in the olden days, and actually still today to some extent, the way you would do screens would be using forward genetics. And here you take an organism, you mutagenize it, you select for some kind of a phenotype that you're interested in, say resistance to a drug, and then you find the gene that's responsible for that phenotype, usually by complementation or something like that. And this was, for a very long time, the way that people did genetics in an unbiased fashion. Of course, you can also study heritable traits and then find genes that way. But this is the way that people have been doing genetics for a long time. With the sequencing of the genome, a number of different technologies have enabled reverse genetics, which is now that you know the sequence of those genes, you can either use, for example, an antisense oligonucleotide or RNAi, which was the technology of choice for about 10 or 15 years, where you can specifically code a double-stranded RNA, a short, I should say, 21 nucleotide double-stranded RNA that effectively incorporates into the cell's endogenous RNAi processing machinery, which in the end causes the degradation of a target RNA molecule. And so this, I should say, has been used for quite a long time, still actually has some uses, as I'll describe to you. But the key idea here is that you're introducing, whether by a chemically synthesized double-stranded short RNA molecule or an lentivirally encoded hairpin that in the end gives you that same product, a double-stranded RNA that causes degradation of its target. So now that we have the ability to knock down specific genes and the sequence of the genome, you can do genome scale reverse genetics, and that allows you to potentially find all the genes involved in your favorite process. So instead of just mutagenizing a plate of worms or bacteria and hoping that you find an interesting mutant that gives you the phenotype you'd like, you can really systematically scan through every possible gene, knock it out or knock it down, and ask what is the phenotype of that gene. And so I say that's easier to saturate because if you wanted to actually saturate the entire genome doing a chemical mutagenesis screen, you would need an enormous vat of worms or bacteria and thousands of people years to actually potentially get all the available mutants that might give you all the phenotypes. But if you know the sequence, you can actually go in and just surgically knock them down or out one at a time. So this is a much more efficient way of associating the gene with the phenotype, and now that we have not just the sequence of the genes, but actually all these regulatory elements in the genome that Luke was describing, we can actually approach systematic annotation of the genome, both for coding and non-coding areas. So there's a lot of information on this slide, but I want to just describe the two general strategies that have been used for doing genome scale screens. So on the right, you'll see arrayed library screening. This entire slide is geared toward CRISPR screening, but a very similar overall workflow was used for RNAi for a long time. And the key differences are that in this case, you have one well, one gene knockdown. And in this case, you have a pooled library where you have lentiviral, usually plasmids encoding either an shRNA or later a CRISPR guide RNA. And these are infected at low multiplicity so that each cell gets one virus in a pool. And then you can subject this pooled population to selection. And so in the end, in both cases, you end up with some selection strategy and then, informatics to separate the hits from the non-hits in these types of screens. One of the nice things about an arrayed screen is that you can potentially get much more high-content imaging. So you can put these plates after you knock down one gene in a well into a microscope, take an image of what the organelles look like, for example, and really get an enormous amount of information for what's actually going on in that one well. You can't get typically that level of information in a pooled screen, but these have a number of advantages. They're much, much easier and cheaper to conduct. So I'll just tell you right now, everything I'm going to talk about from here on out is in pooled format because we love the pooled format. But I wanted to set this up because a lot of screens were done this way, and it's still actually possible to buy even now CRISPR guide arrayed in libraries, because there are some applications where you really want an arrayed one gene at a time, one well at a time screen. Okay. So. Just to kind of go back to the history of these types of screens. So for a long time, screens were done RNAi screens were done in an arrayed format. And typically the workflow it was sort of summarized in the slide before, but essentially you have a stock plate which has one siRNA in each well. You end up transfecting that into the corresponding well of cells through one or another strategy, and then you expose those plates to some kind of drug, for example. So in this study from Michael White's lab, they were looking at modifiers of a chemotherapeutic drug called paclitaxel, and they could find a number of genes, including proteasome, different microtubule kinetochore binding proteins, were modifiers of the toxic activity of this cancer drug. And so the utility of this approach was, can we find genetic modifiers that might be useful in the setting of cancer, where you use a drug like paclitaxel, which is frontline chemotherapeutic in breast cancers, for example. Sometimes that drug fails. Could you find another target that would actually improve the efficacy of that drug? So in some cases these worked. Here's a pooled screen. Now each shRNA is encoded in a single lentivirus. These are infected into cells, and the screen here is to find, in the context of a KRAS mutant cancer, genes which, when you knock them down, further sensitized to another kinase inhibitor, a MEK inhibitor. So MEK inhibitors have been this powerful therapeutic that have been used in some cancers. They almost always fail because the cancer finds another way around it. And so this was a synthetic lethal screen to try to find combinations of second targets that would help a MEK inhibitor to kill KRAS mutant cancer cells, which in general are thought to be undruggable. So KRAS is this powerful oncogene that's mutated in a huge number of cancers. It's thought to be difficult or impossible to drug. So if you could find other targets that would work in a KRAS cancer, you might have a good way of killing these cells. And so in this pooled screen, they infected an entire genome-wide library, knocking down one gene at a time. They treated with this MEK inhibitor at a dose that would kill some of the cells, and then look for things that, when you knock them down, actually further allow killing of the MEK inhibitor. So this is what they found. The MEK inhibitor is called selumetinib, I think. And what they found is that in the presence of a control shRNA, the cancers still grow, but if you knock down BCLX long and you use this inhibitor together, now you have synergistic killing of these cancer cells. So this is to show that whether you're doing an arrayed RNAi screen or pooled RNAi screen, the technology actually works. I'll say there have been enormous improvements in CRISPR screens from work from Luke and work from a number of other labs that I'll describe, where it's possible to really improve the specificity of these screens. Having said that, it's possible to do an RNAi screen and actually get useful results, and I'll describe a little bit more about this. So this was taken further, and they actually took tumors in mice and treated with that same combination of now an Abbott drug that targets BCL-X lung and MEK inhibitor, and it could actually show tumor regression. So just to show that you can take an in vitro screen, use those findings to actually translate it to anti-tumor therapy. Okay, so while there were, shining examples of that type in the literature where you could find something really useful, there were also these truly alarming examples of how terrible RNAi can be. And this is one of the reasons why people have been so excited about how dramatically improved CRISPR screens are. This is sort of a classic example in the literature of 3 different labs who all did an HIV screen. So ostensibly, this is a virtually identical screen. I believe they used the same cell type. It was a screen for resistance for HIV infection. I think they used different RNAi libraries. There were 3 different labs. But what you see here is that the overlap in their findings was actually probably lower than you would expect by pure chance, which is actually sort of impossible if you think about it. But it's one of those cautionary tales that is actually often cited in reviews as an example of how bad RNAi can be. And just to be completely fair to these folks, it's not like they're terrible labs that don't know what the hell they're doing. It's that there are fundamental problems that actually need to be considered, I think, in any screen, not just RNAi screens, but CRISPR screens as well. RNAi is subject to massive off-target effects that are typically not seen in most CRISPR screens, although I'll give some caveats for that in a minute. So Luke showed this amazing figure where you knock down with CRISPR-I GFP, and that's the only gene in the whole genome that gets changed. You would never, ever see that in an RNAi screen, because if you put an RNAi species in a cell, of course, depending on how much shRNA or siRNA you express, hundreds of genes, in addition to the one you're intending to target, also change. So off-target effects are a major problem. With siRNA or shRNA, reagent heterogeneity is a major issue. It's still an issue with CRISPR guide RNAs, so we still don't know what is the perfect sequence of a guide RNA. But this is an even bigger issue with shRNAs. It's actually not that easy to find a good siRNA or an shRNA. At the same time, differences in methods for calling hits, cell lines, and the protocols that are done in different people's labs will always be a source of error, and so that should be kept in mind. I think even now, with the latest and greatest CRISPR libraries and people who are using what ostensibly should be ATCC-derived K562 cells, you sometimes only see 60% overlap between 2 labs that have done a nearly identical screen. So there are some factors that probably won't be solved in the near future that account for differences between labs. Okay, so enter CRISPR. So by comparison to an shRNA screen or RNAi screen where you're knocking down transcription of an mRNA, with the initial CRISPR screens that were done, and of course this is now complemented with the CRISPR-I type screens that Luke has done, you can take, for example, active Cas9, create an indels in genes and effectively cause functional gene deletion. So there's a huge difference now. You're causing an effective gene deletion at the DNA level versus knockdown of transcription at the RNA level. And I should say a lot of the things that I'm describing in the context of RNAi are now applicable and actually even better with the CRISPR-I system. So you might just substitute in some cases shRNA with CRISPR-I when you're thinking about how you might conceivably compare a knockdown phenotype with a knockout phenotype. Okay, so one actually useful figure, this is I think from Feng Zhang's lab, but basically they're showing the type of differences in gene expression you might see in a knockout system versus a knockdown system. So even though there are these nice examples in the literature of when you target a gene with active Cas9 and get nearly complete deletion, in fact, the amount of cutting and editing of alleles you get is quite heterogeneous. So, at best, you're going to get genes that were never edited, some heterozygotes, and some complete deletions. This is in fact much more complicated in some cancer cell lines where you have more than 2 alleles. You may have a real spectrum of different editing phenotypes, and that accounts for some of the heterogeneity. If you saw in the slide that Luke showed from Bruce Conklin's induced nuclease, there's a lot more heterogeneity in that cell type than there is in the CRISPR-I system, and that's partly because you can't really control what happens when you induce editing with active Cas9. This has important implications in a screen context, because you're now looking at a pooled population of cells, and you have to think very carefully about what do I call a hit if my population of 1,000 cells, each of which knocks down one gene, has an array of different alleles being knocked out. By contrast, RNAi or CRISPR-I, while you may have not a complete deletion knockdown, it would at least have a more uniform perturbation. So in principle, you could get an allelic series. So a uniform perturbation and different levels of knockdown. This can actually be useful in some contexts. So this is sort of one of the examples from one of the early screen papers, where you can see here's an shRNA knockdown where you have different levels of knockdown, or CRISPR knockout. This looks amazing, but I should say this is a single allele of GFP. Turns out to be a very easy gene to knock out, because even frame shifts in this gene cause effective loss of fluorescence. But what you'll see, even in this seemingly near perfect scenario, is this population of cells that actually weren't edited at all. So this is something that you need to consider in the context of a genome scale screen. You will have some cells that are not edited. Okay. I should also say that both of these systems suffer from off-targets, and I thought I would just briefly kind of compare and contrast some of the useful properties of each of these systems. So CRISPR in general has greatly fewer off-targets. I have an asterisk there because I'm going to show you that at least for active Cas9, there are some very significant off-target effects that should be considered, especially in the context of a screen. Namely, that if you have an active nuclease in a cell, it can cause DNA damage, and that DNA damage actually has a measurable growth phenotype and potentially can be a modifier of other phenotypes that you're interested in studying. Having said that, if you look, for example, at what Luke had showed for that single GFP knockdown, it tends to be much more specific than RNAi, certainly. It's possible to completely delete genes. It's much easier to find effective guide RNAs. Not only can you delete genes, but as Luke has elegantly shown, you can also activate and repress gene transcription. RNAi, while it has some limitations, as I pointed out, one of the really nice things is that there's a lot less engineering required. So one of the unfortunate kind of pains in the ass about doing a CRISPR screen is you have to get all of those components into the cell. So you have to get Cas9, you have to express it, you have to get a guide RNA, you have to express it at high enough levels. There's a lot of reasons why you obviously want to do that, but I will say one of the nice things about an shRNA screen is you actually only have to put in that one component, and you still have to worry about expression levels, but it's less engineering. Because they don't actually induce DNA damage, even though they have their own toxicity due to saturation of the microRNA machinery and off-target effects, there's less nuclease toxicity in an shRNA screen. The perturbation can be more uniform. It's possible to target specific transcripts, although of course it does have a significant increase in off-target effects. So these are some pluses and minuses to keep in mind. Some of these apply to the CRISPR-I system, less toxic than than active Cas9 for sure, but actually many of these disadvantages are not present in CRISPR-I. So I think increasingly people will be sort of shutting down shRNA screens, although because of this type of thing, in some cases it's actually really nice to be able to pop in an shRNA and not have to worry about getting Cas9 and the guide in. Okay, so in some of the early work that I was doing together with Martin Koppmann and Jonathan Weissman's lab, we actually were at that time building what we had thought was a really nicely improved RNAi system. And so I'm going to just briefly describe this because it actually fed into what we then used to do the CRISPR screens. But essentially, we got around this problem of low on-target efficacy and a huge problem of off-target effects by just making a massively redundant library. So instead of the 2 or 3 shRNAs or siRNAs that people had used in the past, we just put 25 shRNAs. And if you use that many shRNAs, you're bound to have some that will work, and you have enough of them that even if there's an off-target for one of them, you can ignore it because you look at the consensus phenotype of those shRNAs. And this greatly increased our ability to statistically pull a significant significantly pull out hit RNAs in screens. And of course, it's possible because of the two key technologies that enable really a lot of the stuff that we're doing now, the ability to synthesize these pooled libraries in a complex microarray synthesis platform, and the ability to sequence those libraries at the end. So our platform that we built together with Agilent of course, builds on work from Steve Elledge, Craig Hannon, George Church, and many others who had used microRNA-based synthesis to not just make one shRNA, or the exact same thing can be used for guide RNAs, but to synthesize now tens or hundreds of thousands of different guide RNAs or shRNAs in one shot. And you can clone these because the oligos are high enough fidelity directly into a lentiviral vector in about a week, plus or minus. It allows you to do very rapid screening in pooled format. So instead of the arrayed format, which I should have mentioned also comes with an enormous cost and just time to actually screen through those libraries, these pooled screens are quite fast. And importantly for our purposes, the platform is very highly adaptable. So when, after we had spent all of this time building what we thought was a great shRNA platform and the CRISPR revolution happened, we could just take a guide RNA and drop it into that exact same lentiviral vector and effectively use the same workflow to do now CRISPR screens instead of shRNA screens. And because we can also change the algorithms that we use to design these shRNAs or guide RNAs, we just tweak the vector to make double guide RNA platform expression vectors. This platform has been quite valuable for making these changes. So the general workflow for the pooled screens that I'm going to spend most of the time talking about is we build a pooled lentiviral library that code the guide RNAs. We infect them into cells so that each cell gets one guide RNA. These are typically already expressing Cas9. We then do a pooled screen, and I'll describe a number of different pooled screens that you can do, but a standard option that we might do is to look for modifiers of a drug. I showed you two examples of different cancer drugs that have been used. So we do a lot of these in my lab, but a number of other labs have done this for a long time to look for genetic modifiers of a drug. And in the end, you sequence these populations, and you compare the treated population with the untreated population. You count the abundance of the guide RNAs, and you can call hits. In our libraries, we use a huge number of negative controls, so you can actually call hits. I think there's another lecture on statistics of calling hits and things like that. So I'm not going to spend too much time actually describing how the hits are called. But suffice it to say, for most of the libraries that we use, we have a huge number of negative controls that will allow us to carefully define what no phenotype looks like. And then you can compare the phenotypes of the gene guide RNAs targeting your favorite gene to that population and very clearly call hits from non-hits. At the end of the talk, I'm going to describe some further work that we have done to take the output of this type of screen and actually do genetic interaction measurements. So at the end of a screen, you might have 100 genes or 500 genes, and you would love to know, well, what do these things actually do? So we've come up with a strategy to repurpose tools that have been built in yeast to do systematic pairwise interaction maps and actually understand gene function in pooled format. And I'll describe that toward the end. So some typical pooled screens that you might do and considerations. So as I mentioned, a standard kind of workflow is to introduce a library, take your favorite drug, for example, and then look for modifiers of cell death. This tends to be incredibly easy to do. And so a lot of screens that people do out of the box are just growth-based screens, where you're looking for things that either increase or decrease the growth of a cancer cell by itself, or modify the activity of a drug that allow you to do these sorts of screens. A number of other strategies have also been employed. So you can, for example, follow the activity of your favorite transcription factor by coupling it to either a fluorescent and you do flow cytometry-based screens, or you can couple it to expression of a drug resistance marker, and then do viability-based screens that are reporting on the activity of this promoter. So your favorite stress response, for example, you could couple to these different promoters and actually do sorting-based screens based upon those pathways. And so I think one kind of practical tip that's useful is if you're doing these types of screens, it's useful to have a counter screen where you just look for modifiers of a general transcription or translation factor. So in addition to this, you might consider having just a constitutive promoter that drives mCherry expression. And then you could subtract everything that affects just transcription or translation of mCherry as what you might call background in that screen, and then only look at things that specifically modify the expression of GFP. So this is one probably a useful constraint if you're thinking about these types of pooled screens. If you're doing these sort of depth screens, we in my lab, for example, have found about 50% killing works quite well. You can find modifiers in both directions, but depending on the type of screen that you're thinking about doing, sometimes people will use 90% killing, this sort of apocalyptic death model where everything dies except the 5 genes that are absolutely critical for a process. That can be a really useful strategy for, getting rid of a lot of noise. On the other hand, you can lose a lot of things that way. So other people will screen at a 10% kill, and then you have much more dynamic range for things that sensitize to the drug. So depending on the particular paradigm that you're interested in, whether you're looking for synthetic lethal combinations, whether you're looking for critical transporters for the entry of a drug, for example, different levels of killing might be useful for those types of screens. Okay, I'm just going to briefly go over this slide because it's only to make one point, which is that depending on the type of cell line that you're using, a very important consideration is just the promoter or the vector system you're using to express an shRNA, Cas9, or guide RNA. We have collected from ourselves and a number of other folks, including Luke, who's been incredibly generous with sending us some of these expression vectors, just an array of different promoters that have been quite useful because cell types differ enormously in the extent to which they will reliably express guide RNA or Cas9. And so one of the biggest issues in actually conducting these screens is establishing a cell line that reliably expresses Cas9. And so, I would suggest if you're actually thinking about doing this, talk to someone like Luke, who has an enormous amount of experience in doing this, or myself or a number of other people. And potentially just test a number of different constructs because actually trial and error can actually help enormously in finding an effective system for just expressing Cas9 in a guide. Okay. So those are sort of practical things to keep in mind. One other practical consideration I thought I would just mention is the importance of maintaining library representation. So these pooled libraries, for the shRNAs, we had 25 shRNAs per gene. Fortunately for CRISPR guide RNAs, it's possible to really shrink the size of those libraries because it's much, much easier to find an effective guide RNA. And with the improved algorithms that Luke has described from Max's work, for example, you can now shrink the library to maybe 5 guide RNAs. Some people are even trying 1 guide RNA, which I think is kind of ludicrous, but in principle, there's so much easier to find effective guide RNAs that you can really shrink the size of these libraries. Having said that, it's actually quite important to maintain effective representation of these libraries. So when we do a screen, we typically try to maintain 1,000-fold representation. This is the noise you see between 2 replicates at 1,000-fold. Actually not too bad. But if it were possible to improve that coverage to like 50,000-fold, you can see the noise goes almost to 0. So it's something to keep in mind that the amount of coverage you get actually can really influence the noise in these types of screens, even with a really improved CRISPR library. Okay. So I'm now going to describe some of the genome scale screens that have been done. A few of the first ones with CRISPR were using active Cas9 and were conducted by David Sabatini's lab, Feng Zhang's lab. And what they basically did was to take an oligo design, which in effect incorporated some of the same rules that Luke described in terms of optimizing guide RNA properties. A lot of this stuff has been published, and effectively targeted what were conserved exons, and they thought to go as 5' as you could, with the idea that they wanted to make a guide library that would work on as many different transcript variants as they could find. And if you put a stop codon as early as possible in the gene, you had the best chance of actually effectively deleting that gene's function. So in general, this worked pretty well. This is sort of a V1 library, and I'll show you some other examples where some of these rules have been tweaked and improved upon. But the idea was you take an oligonucleotide synthesis platform, you generate a genome scale library. I think in this case they did maybe 3 to 6 guides per gene. And then the workflow is basically exactly the same as what I described. You have a lentiviral library. Each vector codes for one guide. You infect it into cells, and then you can do a screen. And so one of the first screens that they did was actually just a growth screen. So this is just effectively called a screen for gene essentiality. You want to find all the genes that are required for growth or viability in a cell. One of the interesting things you can see in this plot, so here you're seeing the ratio of cells compared at day 3 to day 14. So this is growth over a 2-week period. And what you can see is that you now, after 14 days of growth, start to see this population of cells that are lower in abundance than they were at day 3. And that is because they're dying, of course. So you knocked out a gene that was important for their growth. You can see what some of these genes are. They're key cellular processes, RNA processing, RNA binding, ribosome biology. These are core machineries that are absolutely required for cell growth. The interesting thing actually is that it's much easier to break something in a cell than it is to make it go faster. There are very, very few genes which, when you knock them out in a cancer cell, actually make those cells grow faster, compared to the thousands of genes, actually, depending on how you draw the threshold for a growth phenotype, that actually break or significantly impair cell growth. So this was nifty. One of the other things that they did in this paper was to do a drug resistance screen. So I just showed you a growth screen where you're knocking out a gene and you're just looking at across time, at an early time point and a late time point. In this screen, what they did was they put the library in, and then they treated with this BRAF inhibitor that normally kills the cell, except if they knock out these genes, which it turned out are different, direct members of this pathway, or genes that had been previously shown to be cause resistance to this BRAF inhibitor. So one of the cool things that they highlighted in this work was that all 4 of the guide RNAs targeting this gene, for example, you're right, look, it's totally impossible to see this thing, but NF2, for example, all 4 guides actually show the same phenotype. They're considerably higher in the treated cells than they are in the untreated cells. And this is something you would basically never, ever see in an shRNA screen. So you would never see all 4 shRNAs giving you the same phenotype. Here you see all 4 guide RNAs cause significant resistance. And the other interesting observation is you don't have 1,000 genes that are required for HIV resistance. You have 8 genes or 20 genes or something like that. So you have a really narrow list of almost certainly right hits, and when they actually go in and validate these, 90+% of them validate. So I think it shows the power of these screens to get really clean phenotypes for gene knockouts. One caveat that I thought I would point out from our own work is that if you looked at this, you might say, well, all 4 of these guides are working really well. In fact, I just said that. But in fact, there's a lot of heterogeneity in the performance of those guides. And you don't really see that until you really look carefully at the phenotypes for all the different guide RNAs, not just in a resistance screen where you're really driving all those things to cause an increase in growth, but in a dropout screen. So in this plot, what we're showing is actually the phenotype of really critically essential gene, ABL. BCR-ABL is the gene that drives K562 cells. If you knock it out, they're dead as a doornail. And what you can see here is that of all the guide RNAs that target it, there's a huge range in effectiveness. Some of them, kill it completely, and some of them have a pretty mild phenotype. And so it's important to consider that with any screen, you're going to have a range of guide RNA phenotypes. Okay. So one of the other things that we've done that's been quite useful with these types of screens is to actually do a drug target identification. So I showed you a couple of examples where people had looked for modifiers of a drug. In most cases, those drugs already have a known target. So they're targeting some kinase, which is thought to be really important for cancer cell growth. And you want to find a second target, which is important, for example, for a potential synergistic target for that drug, or to understand how that particular cancer works. Another useful application of these screens has been to identify de novo the target of a drug that does something interesting, but you actually have no idea how that drug works. So in this paper together with the Cleary Lab, who had done a chemical screen. This is actually quite common in the pharmaceutical industry. People do giant chemical screens. They find some drug that magically kills leukemia cells in this case, but not normal cells. So it looks extremely interesting, but they have no idea actually how this thing works. What we were able to do is to take an shRNA library, and this is actually just a half genome screen, and systematically knock down one gene at a time, and with the idea that if you knock down the target of that drug, now that cell is much, much more sensitive to the drug than all the other cells. So you can see with actually pretty remarkable, actually maybe being too remarkable specificity, we were able to pull out what happened to be the molecular target of this drug, which is this NAD biosynthesis gene called NAMPT. You might say, why didn't you find all the other NAMPT genes? One thing I should say is we probably should have just stopped doing screens entirely at this point. It was a really nice result. It's a half genome screen, and probably because this was an early shRNA library, we had bad shRNAs for the rest of the components of that pathway. But it does illustrate the general principle that you can use these genome scale screens or near genome scale screens to do unbiased drug target identification if you don't know the target of a drug. So in another example of a similar type, in my lab we compared, we actually tried to find the target of this drug. This is now an antiviral drug which targets the host cell. So there are a lot of different antiviral drugs in the world. Typically they target the virus itself, and so if the virus mutates, you just need to find a new drug for that new virus. This is a pain. And so if you were able to find drugs that actually target a host process that cause a protective phenotype against a range of viruses, that could actually be quite useful. And so we set out to actually identify this target, which had been identified by GlaxoSmithKline in a similar, large-scale chemical screen for compounds that inhibit the replication of a range of different viruses. So they found this thing, GSK983, that looked like it was effective at inhibiting adenovirus SV40 and HPV16, they actually reported a kilogram scale synthesis of this compound. So this is another common thing that happens in pharmaceutical industry. They do a lot of work, probably millions of dollars worth of research on this drug. They synthesize kilograms of it, which means they're testing it in animal models, and then inexplicably they just drop it, and they have no idea what the compound did. Typically that's